Hello, my beloved students. Okay, have you ever thought or have you ever been thinking that one day there will be a organism that can glow in the dark? Yeah, so you can see from this diagram, Andy. Alright, Andy was born as the transgenic monkey in 2000, right? Okay, how does Andy can glow in the dark? Okay, in normal day, Andy is just like a usual monkey. But when at night, Andy shows the green color. So how Andy get this green fluorescence color, right? So it's actually uh, Andy carry a harmless gene for gene fluorescence protein, that's GFP, okay, into the Andy's research genome, right? So another situation, right? Uh, Nice, right? Okay, you can see the fish can glow in the dark. Okay, example, right, like freshwater zebra fish. Okay, these fish, how they can glow in the dark? Okay, with the glowy orange, okay, glowy yellow, glowy purple, my favorite color. Okay, by integrating a fluorescence fluorine protein gene, okay, from the jellyfish into the embryo of the fish. Wow, right? So why? This is for the good of water quality, to detect the water pollution. Is it nice, right? You can see the fish, okay, real fish that can glow in the dark at night, right? Okay, another unique, right? If you ever think that tobacco plant also can glow like a fireflies? Yes, it's not impossible nowadays. With the technology, okay, we can integrate or we can recombine the DNA of gene fireflies into the tobaccos, and we can produce what we call as a transgenic organism. So how does it can happen? So the term that teacher would like to introduce is R, the small r, okay, DNA. So this is our self, right, human, compared to the prokaryotic, that is bacteria. Okay, so why bacteria is so important in this our discussion today? Because actually bacteria is the organism, right, that will play a big role in formation of rDNA. Okay, what is R class? R means stop your recombinant DNA, right? So actually chapter 8, okay, we are going to learn about how recombinant DNA is produced from by the technology. Now this technology, okay, as you all know, really important in our life, right? So there are three learning outcomes. Okay, number one is the recombinant DNA technology itself. Number two is messenger cloning, and number three is the application. So we are going to define first what is the recombinant DNA. Like teacher introduced now, okay, the small r and DNA. So it means that DNA that contains gene. Gene of what class? Gene from different sources that have been combined to the techniques that we call as a genetic engineering. And when we mention genetic engineering, it's actually the process of changing the genetic makeup. Okay, teacher repeat class. Genetic engineering is your ability, you engineer the DNA by how? Changing the genetic makeup of the DNA, right? So this is what we're going to learn class. Genes, technique genes, two different sources. Okay, combine in vitro, where you're going to do this in our laboratory class. Okay, there are five tools in gene cloning. Number one is target gene. Number two is restriction enzyme. Number three is DNA cloning vector. Number four is modified enzyme. And number five is a host cell. But in this video, teacher will just like to emphasize on the number two, the restriction enzyme. Okay, so this one in class, where restriction enzyme is actually enzyme that used to cut or to cleave the DNA into fragments, right? So tools in gene cloning restriction enzyme. So basically, restriction enzyme is an endonuclease. Meaning what class? Endonuclease is one of the classes of enzyme that produce in the cell, okay, inside the cell. And this enzyme able to recognize the first point, class, the first point of restriction enzyme is recognize. Recognize what? Recognize restriction site, and after they have recognized the restriction site, they are able to cleave the DNA, okay, at the molecule specific nucleotide sequence, which is restriction site, and able to split the DNA into a fragment. Okay, okay. Basically, class restriction enzyme in daily life of the bacteria okay, is used to protect the bacteria cell. Right, so bacteria cell, right, will secrete its own restriction enzyme. That's why you call it endonuclease enzyme, right, to destroy the pathogen or all over what over foreign DNA that invade their body, right? So that is how the restriction enzyme works in the bacteria. Okay, now we are going to use this restriction enzyme, okay, artificially or during using it in the technology. So how it works? So but before that, teacher would like you to get familiar this term, that restriction site. Okay, restriction site is actually referring to DNA sequence, that cleavage site that cut by a particular restriction enzyme. Okay, so as you all know, class, restriction enzyme as a protein, properties of enzyme, they are very specific. Okay, they can able to recognize a particular short DNA, okay, which we call restriction enzyme. Okay, so restriction site basically 4 to 8 bases. Teacher can repeat the class. 4 to 8 bases, right? For the restriction enzyme to recognize. Right, so what is okay, before we proceed class about the restriction site, teacher would like you to read these words, okay, forward, okay, from the frontward and the backward. Okay, what can you say about these words, class? Okay, from backward, okay, and from frontward, okay, these words give the same meaning. Okay, civic, from backward also civic, Hana, kayak, level, madam, okay. Alright, so now what is actually referring to palindromic? Okay, palindromic, just now is palindromic words. Now teacher now introduce you a palindromic sequence. Okay, palindromic sequence is actually referring to the same base sequence in opposite direction on both DNA strand. Okay, teacher repeat again. Palindromic sequence mean the same base sequence in opposite direction on both DNA strand, and it's only a short DNA segment which consists 4 to 8 bases. Okay, 4 to 8 bases sequence, right? This what is called a palindromic sequence, act as a recognition site, okay, for the registration enzyme to cleave. Right, so this is the example that given, right? So let's introduce you to two type of restriction enzyme, how they cut at the restriction site. Okay, first we call it a sticky end. So sticky end is actually one way how restriction enzyme, okay, cleave the DNA and produce a staggered cut. Okay, cikgu repeat the class. Okay, teacher repeat class. Staggered cut. Alright, so you must familiar with this term class, staggered cut. Alright, so this staggered cut is actually meaning they will expose the single strand. Okay, and okay, they so we call it a sticky end. Okay, this staggered cut, okay, will form hydrogen bond with compatible sticky end of another molecule of DNA, which also cut with the same enzyme. So how this is how the restriction enzyme works, right, class? So these are the example that teacher would like you to have an idea in your syllabus. Okay, so there are two types of restriction enzyme. Okay, one we call it EcoR1, and number two is a BAM-H1. Okay, these two enzymes we extract. Okay, we get it from a bacteria. So based on their name, EcoR1 is actually we extract from Escherichia coli. Right, and the base sequence or the restriction site that involves is 6. Okay, 6 class. So which one? This is the first one where you can read the GAATTC. Okay, teacher would like to introduce you to the acronym, okay, of the this restriction site, okay, which is palindromic. Alright, so gua, okay, anak, ayah, terbaik, tetap cun. Okay, that's the way how you nak memorize, right? So from okay, from frontward, okay, and from backward also, right? Gua, anak, ayah, terbaik, tetap cun. That's how you memorize, okay? Guanine, adenine, thymine, cytosine. Alright, so for BUMP H1, I leave it to you. So then later you will share with me how you memorize it according to acronym. Okay, next, this is how it works. The diagram shows. So this is what teacher mentioned just now. Okay, sticky end, which involve staggered cut, right? Okay, so this is another example of sticky end cutting by BUMP H1. Okay, which also produce staggered cut, or we call it a sticky end, right? Okay, another type of cutting, okay, or cleaving of restriction enzyme, okay, is cut by simple cut, right? So simple cut across both strand a single point. Okay, so it will produce, okay, the cut as a blunt end. And one of the example that you must remember is SMA1, where we extract this enzyme from bacteria that known as Ceratria mercensens. Okay, and the sequence is also involve 6 bases and easy to remember, class. So triple C, triple G, okay, frontward and backward, triple C, triple C, triple G. Okay, and how the small one cut? Okay, here will be small one. Okay, small one will cut here and produce blunt end. Okay, so this is how it works. Class, small one cut through triple G. So this green color we call it a restriction site, which must palindromic. Alright, class, I say palindromic, so it will produce two blunt end. Okay, two blunt end, simple cut. Okay, class. Alright, so the unpaired base of DNA fragment can form hydrogen bond with compatible of the sticky end of the another fragment. Okay, so that's all for today, class. Thank you. See you again for our next video. Bye.